Okay. Hello, everyone. Today is my great pleasure to introduce our today, Ping-Chih. Ping-Chih is a cancer immunologist with a particular research interest in immunometabolism, the tumor microenvironment, and the viral infection. So I think the ultimate goal for Ping-Chih's research is to exploit the findings to better harness the immune system to fight against infection and malignancy. And also, I think Ping-Chih has the vision to reprogram the tumor microenvironment, reverse immunosuppression, and broaden and boost the efficacy of cancer immunotherapy. So just a little bit of background about Ping-Chih. Ping-Chih grew up in Taiwan and obtained his basic biomedical training, including both a bachelor degree as well as a master degree at National Taiwan University. He then obtained his PhD at the University of Minnesota. Ping-Chih then did his postdoc training with Susan Cage at Yale University, where he demonstrated how cancer cells evade T-cell immunosurveillance by depriving the infiltrating T-cells of glucose, which is consumed by large amount by malignant cells. In 2015, Ping-Chih relocated to Switzerland as a tenure-track assistant professor at the University of Lausanne. And there he actually had a very successful career as an independent PI. So Ping-Chih was very soon promoted to tenure associate professor in 2019. And then he's also promoted to associate member at the Ludwig Institute for Cancer Research in 2020. So Ping-Chih's research is also acknowledged by many international awards, including the CRI Lloyd Old Star Award, the Embro Investigator, Young Investigator Award, the Melanoma Research Alliance Young Investigator Award, and established the Investigator Award, and also the Swiss Bridge Award. So on a personal note, Ping-Chih has been a great friend and colleague to me for all these years. And I remember when I was still a graduate student at Yale, Ping-Chih really gave me tremendous help to get started in this exciting area of tumor immunology. So without further ado, I welcome Ping-Chih. I've always been very impressed by your energy and your enthusiasm. So I really look forward to your talk today.
Okay, so Chen Chen, thank you very much. So I assume everyone can see my slide, right? Okay, so then I'll just go ahead. So first of all, it's my pleasure to have the chance to share our work with you, and then also have the chance to interact with the old friend and also new friend. And in this format, I think it's also very important for us. It's also very important. Okay, it's also very important for us to disseminate the work we did in the past few years. So, but today I will talk about the how the mitochondrial dynamics can instruct T-cell immune response in a tumor microenvironment. So I will combine two studies we published recently, and to give the overview how this will be how this can be a very important regulation in the immunometabolic angle for controlling T-cell immune response. And so please feel free to let me know if you have any question, and also I will be happy to answer if you have any particular question, and also the following study we might go ahead based upon those findings, and I will be happy to answer your question back. So okay, so since we talk about the T-cell anti-tumor response, so we basically need to consider how we can make the T-cell do much better in the tumor microenvironment, how we can win this battle by using our immune system for fighting with malignancy. But if you think about the entire equation, it should be very simple if you want to achieve a good anti-tumor response. So one of the features you can do is that you can you just need to bring in your T cells, which can recognize tumor antigen or a neoantigen present by those tumor cells in a more efficient way. At the same time, once those cells enter to the tumor microenvironment, enter to the battlefield, you also need to feed those cells well. If you can achieve these two steps, you will be able to launch a very proper and very strong anti-tumor response. However, the microenvironmental challenge is on the right-hand side. Can you hear me now? It looks like I was mute by some host.
Yeah, go ahead. Sorry.
Okay, it's okay. Okay, so now you can see my screen again, right? So those microenvironmental challenges, including metabolic restriction, hypoxia, and also the poor T cell infiltration, engagement of co-inhibitory receptors, as well as the accumulation of immunomodulatory cells, including regulatory T cell, MDSC, M2 macrophage, and tolerogenic dendritic cells. They represent different layers of regulation to shut down our host anti-tumor immune response. And today I will talk to you about the first one and third one and try to let you know how the mitochondrial dynamics in T cells can control their property in terms of metabolic fitness and also in terms of the accumulation of immunomodulatory cells in the tumor microenvironment. So the first one I want to talk to you is about the accumulation of regulatory cells in the tumor microenvironment. So we know that regulatory cells, so-called Tregs, they play a very important role to control tissue and immune homeostasis by controlling the activation of macrophage, dendritic cells, CD4 and CD8 T cell, as well as the endothelial cell growth and the structure formation through multiple ways. And this is just the summary of their actions in terms of what I just mentioned. We published in 2018 in the review for Trends in Cancer. So I will not go through the details. I just want to give you the flavor. Actually, regulatory T cells, they can play a very critical role to do that. So this is a typical role we consider Tregs can do. However, in the tumor microenvironment, we also know that you will see robust increase of intratumor Tregs. They just stay in the tumor microenvironment, and people also notice that once you have more regulatory T cell accumulate in a tumor microenvironment, your entire tumor microenvironment will become more immunosuppressive. You will have less ongoing host anti-tumor immune response. At the same time, the prognosis for those patients with high Treg accumulate in the tumor microenvironment is also very low. So basically, with this information, people start to speculate, maybe we should find out a way we can destroy those intratumor Treg, and we will be able to unleash host anti-tumor immune response in a much stronger way. And this is the hope, because based upon the animal model, we know that this is doable. However, when we talk about this kind of treatment, and we have to consider what I just mentioned in the previous slide, regulatory T cells, they play a very critical role to control tissue and immune homeostasis. So when we try to target Tregs in tumor microenvironment, you will start to see those patients receive those treatments. They will have a lot of autoimmune-like symptoms or side effect. This is basically due to systemic impairment or deletion of Tregs. This will result in perturbation in the immune and tissue homeostasis. So based upon this information, we can know that Treg targeting approach is very important. However, the therapeutic index is very, very low due to those complications I just mentioned. So we have to find out treatments that can more selectively demolish intratumor Treg while keeping Treg in peripheral tissue intact and function. So when we start to think about this entire idea, how can we achieve that? We start to have a very interesting guess. So my student told me that maybe we can think about the chimera idea. So for those Tregs, they stay in different places. They will encounter different surrounding cells. They will try to do different things based upon the tissue context signaling. So in this case, they probably need to change their metabolic preference. This is similar to the concept, if I travel to Asia versus I travel to the States, or I stay in Switzerland, I'm still the same person, but I will change what I can eat based upon which kind of food is available in the environment I stay. So maybe Treg, they would do the same thing. So maybe in skin, they start to use very specific nutrient they can get from skin. However, in the tumor microenvironment, they will start to get something which is very abundant in the tumor microenvironment, such as lipid and lactate. Because in that environment, the metabolic stress and metabolic waste and also nutrient can be completely different from other tissues. So if this is the case, we speculate those Treg will engage metabolic adaptation to change their preference based upon where they stay. And we might be able to target this adaptation to selectively destroy intratumor Treg while keeping Treg in peripheral tissue intact and functional. So in order to understand whether this is the case, we decided to learn from patient samples. So we reanalyzed the data published by Sasha Rudansky in 2016. In this study, they isolate Treg from breast cancer patients from both tumor and PBMC. And they did RNA sequencing for those patients that collect and focusing on chemokine receptors, which can be selectively expressed by intratumor Treg. However, my student Hai-Ping did this analysis from the different angles. So we try to see whether intratumor Treg, they will selectively increase particular metabolic enzyme compared with those Treg in PBMC. And when we perform this analysis, we see that most of the enzymes which will be selective upregulated on the right-hand side, as you can see here, this is upregulated in intratumor Treg, are those enzymes controlling lipid metabolism, including binding, oxidation, and biosynthesis. So we start to speculate maybe intratumor Treg will engage particular metabolic lipid metabolism to support their survival, retention, even function in the tumor microenvironment. So we also pay attention to whether there's any particular transporter, lipid transporter can be selectively expressed, increased in intratumor Treg. And when we analyze those things, we first notice that CD36, this is a lipid transporter, a scavenger receptor, which can control medium and long-chain fatty acid uptake, and also the uptake of oxidized LDL. It can be selectively increased by intratumor Treg in a significant amount. And we also observe that FABP4 and 5 also selectively increase in those intratumor Treg. So we start to speculate maybe this is one of the transporter intratumor Treg need to increase. CD36, they have to increase this protein, and then they have to use this protein to get lipid from outside. So we first confirm actually intratumor Treg indeed express higher level of CD36 by comparing Tregs from other tissues from the same tumor-bearing host. As you can see here, when we check CD36 expression in those Tregs, the intratumor Treg express the highest amount compared with draining lymph node, non-draining lymph node, spleen, thymus, and skin from the same host in a mouse model. This is also holding true if we check patient samples. So the bottom part is the patient samples we got from melanoma patients. So if we check those melanoma infiltrating lymph node versus the PBMC of the same host, same patient, you can see that those Treg from the tumor invading lymph node, they have more CD36 expression. And on the right-hand side, this is a pair sample. Each line indicates a pair sample between PBMC and TILs from the same host. So most of the patient we analyze, you can always see this increase. They start to have higher level of CD36 expression in those intratumor Tregs. So with this information, we start to speculate maybe the increase of CD36 messenger RNA level we observe in a breast cancer patient sample analysis, indeed it's true. They will start to upregulate the protein expression as well, which can control their lipid metabolism and how they use lipid, which can be assessed from the surrounding environment. So in order to confirm CD36 expression is quite important for those intratumor Tregs, so we generate a Treg-specific CD36 knockout mice by crossing the FOXP3 YFP3 mice with CD36 FLOX mice. In this case, the expression of FOXP3, which is a key marker of Tregs, would drive the recombinance expression, and this would lead to CD36 genetic deletion in Tregs. So we also use FOXP3 YFP3 mice as a wild-type control, because in this case, they still have the intact CD36. We first confirm when we engraft tumors into those mice, in intratumor Tregs, those CD36 deficient intratumor Treg will have lower lipid content, lipid uptake, sorry, and also lipid content. So it suggests that the expression of CD36 in intratumor Treg indeed can control whether those Treg can get lipid from outside. And importantly, due to the time I didn't show you the data, we also confirmed that if you check spleen and lymph node, those Treg between wild type and knockout, they have a similar level of lipid uptake and lipid content. So the data suggests that only intratumor Treg will rely on CD36 to do this job. And when we analyze this data, we also realize that if we get rid of CD36 in intratumor Treg only, we already can see slower tumor growth kinetics. In the end of the experiments, the tumor burden is also much smaller in those mice they don't have CD36 expression in Tregs. So this result suggests that targeting CD36 in Tregs can selectively destroy intratumor Treg lipid content, lipid uptake, and also may change the host anti-tumor immune response in the tumor microenvironment. So back to our original hypothesis, we speculate we can find out some metabolic adaptation used by intratumor Treg, and then we can target this process to selectively destroy intratumor Treg. So we decided to take a look whether this is the case when we get rid of CD36 in Tregs. So as you can see here on the right-hand side, if those Tregs that don't have CD36, we can already start to see the reduction of Treg abundance in tumor microenvironment. Importantly, in spleen and draining lymph node of the same tumor-bearing host, they have very similar Treg abundance compared with the wild-type mice. So basically, this result suggests that get rid of CD36 expression in FOXP3 positive Tregs, you will selectively destroy intratumor Treg to 40% reduction. However, this is something we also need to confirm because you see that in spleen and lymph node, they have the same abundance. Doesn't mean those Treg without CD36 expression, they still can do their job in the peripheral tissue. So in order to confirm CD36 in peripheral tissue, they don't really need CD36 to maintain their suppressive activity. We perform a T cell transfer colitis experiment. In this case, we transfer naive CD4 T cell into REC1 knockout mice. Due to the lymphopenia condition, CD4 T cells start to expand, and this will create colitis, and mice will start to lose their body weight. As you can see here, their colon and small intestine also start to have a lot of inflammatory signs. And this can be prevented if we co-transfer naive CD4 T cell with Treg from wild-type mice. And importantly, if we transfer Tregs from CD36 deficient mice, we also can see similar level of reduction, prevention of colitis incidence. So this result suggests that in the colitis condition, Treg can still do their job to suppress the ongoing inflammation if they don't express CD36. And we also perform aging experiment to confirm that those CD36 deficient mice, they will not have autoimmunity if they're getting older. So basically, our data suggests that only intratumor Treg, not peripheral Treg, needs CD33 mediated metabolic signals to support their immunosuppressive activity and their abundance in the tumor microenvironment. So we next want to understand why a lipid transporter can control the abundance and the suppressive activity of Tregs in tumor microenvironment. And we try multiple things, including proliferation, including migration, and tissue residency. We didn't see any difference. So we use a very traditional way most people will use, if you cannot find an answer. So we just get the cells out from wild-type knockout of the intratumor Treg, and then perform RNA sequencing and see what kind of signature we can observe. So when we did that, actually we start to see the entire picture become quite clear. So basically, if we compare those intratumor Treg from wild-type versus CD36 knockout, you will see that those CD36 deficient intratumor Treg upregulate a lot of genes are related to apoptosis. And this can be further confirmed if we perform cleave caspase-3 staining or annexin-5 staining, which indicates the cell they are undergoing apoptosis. So you can appreciate that in the intratumor Tregs, if they don't express CD36, they start to have higher level of cleave caspase-3 and also higher level of annexin-5 staining. So basically, our data suggests that if you get rid of CD36 in Tregs, you will start to see 40% reduction of Treg in the tumor microenvironment. And the remaining Treg, they are dying. So basically, they are in a condition they will start to lose their fitness and cannot deal with their survival need in the tumor microenvironment. So we next try to understand why this lipid transporter can control this process and whether the lipid metabolism is quite important to support the intratumor Treg survival and retention. And we realize the mechanism based upon this electron microscopy analysis. When we isolate those Treg from wild-type mice or CD36 knockout mice from spleen and tumor, we start to see a robust difference. As you can see here, when we check those splenic Tregs, between wild-type and knockout, they have very similar mitochondrial number and crystate structure. However, if you check the same tumor-bearing host, but you just isolate the intratumor Treg from wild-type and CD36 knockout, you will start to see different picture. Those intratumor Treg, they don't express CD36, they start to have lower mitochondria number, and they also lose their crystal structure because their crystal become very blurred. So basically, you can see the right-hand side, this is the data, the quantification data of this EN. we can conclude that CD36 expression in Treg can support the mitochondrial number and mitochondrial structure maintenance for those Tregs when they stay in the tumor microenvironment. And you may wonder why mitochondria is important. So I mentioned in the beginning, in the tumor microenvironment, there are a lot of metabolic stress due to, for example, hypoxia, low pH level, lactate, and nutrient deprivation. And mitochondria actually is a very important organelle for every single given cell if they want to maintain their metabolic flexibility. Because mitochondria can use their TCA cycle to control whether the cell can use glucose, fatty acid, or amino acid as a nutrient source to support the ATP production. So once those Tregs that don't have good mitochondrial fitness in a tumor microenvironment, we can start to speculate they may start to die due to the decline of metabolic flexibility they will encounter in the tumor microenvironment. So we start to take a look whether this is the case. So we generate those Tregs in vitro. So we have a wild-type Treg and CD36 knockout Tregs. And we culture those Tregs in normal culture medium or in the same medium, but we give them hypoxia or cancer cell conditional medium, which is the medium we culture cancer cell for 24 hours. And then they contain a lot of lactate and low glucose. If we give these cells, Treg, this kind of medium, similar to the hypoxia, you will start to see CD36 knockout Treg lose their viability. So more of the CD36 knockout cells just die in this 24 hours culture. And the common factor between the hypoxia and cancer cell conditional medium is lactate. So we start to speculate maybe those CD36 deficient Treg, they die due to the high lactate level in this culture condition. So we decide to take a look by using different designs. So we give the cells, those Tregs from wild type and CD36 knockout mice, the same culture medium, but we increasing the lactate level we put into the culture. After 24 hours, as you can see, the more lactic acid we add into the culture, you will see more severe viability issue for those Tregs that don't express CD36. So basically, our data suggests that Tregs without CD36 expression, they lose their mitochondrial fitness and they start to die in the presence of lactic acid, which is quite common in the tumor microenvironment. And you may speculate why whether these two things link together, how the mitochondrial activity can control the survival of those cells in the presence of lactic acid. And this is quite simple. So I have to bring you back to the biochemistry textbook. So in the biochemistry textbook, we always learn that there's a TCA cycle in the mitochondria, which determine the metabolic preference of the given cells, right? But we also know that if we culture cells in a lactic acid enriched condition, cell will die. This is quite similar because, for example, if you do the cell culture, the medium becomes yellow. And if you forget to change the medium, no matter you culture cancer cells or immune cells, those cells will die. The reason is that those lactate accumulate in the medium will make the entire medium become yellow, and those cells die in the yellow medium because they encounter too much lactic acid in the condition. The reason Treg can survive in the lactic acid enriched condition because they can use a very special metabolic program. They convert lactic acid into pyruvate. But you can also imagine that if they want to stay in this condition, like a tumor, long enough, they will have a problem because they will run out of their NAD and generate too much NADH. In this case, if they want to sustain their survival, they have to use mitochondrial activity, because in mitochondrial activity, there's a process called electron transport chain complex 1, which is NADH dehydrogenase. It can convert NADH to NAD. So if you have this process ongoing, those cells will be able to resupply NAD to facilitate lactate become pyruvate, and then cell will not be able to will not be killed by the lactic acid enriched in the cytoplasm. So that's what Treg can do in this case. So they also realized, so we also, in order to confirm those Treg that don't have CD36, they start to decrease their mitochondrial fitness, they will change this ratio and they will start to die because they cannot sustain this entire cycle to facilitate lactate conversion into pyruvate. We first check the NADH versus NAD ratio. As you can see here, we confirm that those Treg without CD36 expression, they lose their mitochondrial fitness, they decline their mitochondrial content and crystal structure. At the same time, they have more NADH, less NAD. So basically, they will not be able to regenerate NAD to support this entire cycle. And we also confirmed that the viability issue for those Treg without CD36 in the cancer cell conditional medium can be reversed or can be abolished if we artificially give those CD36 knockout Treg nicotinamide riboside, which is the precursor of NAD. So we artificially give them more NAD. They will be able to survive in the presence of lactic acid. So basically, our data suggests that CD36 will be able to provide a metabolic adaptation which support the lactic acid usage in the Tregs. So for the sake of the time, this is the first part of my talk. I want to give you the idea how we identify CD36 can be a very interesting target to allowing us to target intratumor Treg. So in the tumor microenvironment, those intratumor Tregs start to upregulate CD36. So they will use CD36 to get fatty acid from outside, but they don't use fatty acid as a nutrient source. Instead of that, they use fatty acid to activate PPAR beta and delta, the same protein. This is very important to control mitochondrial biogenesis and mitochondrial activity. As a result of this action, those Treg with CD36 expression, they will have a very strong mitochondrial activity, which can sustain the lactic acid usage and facilitate the lactic acid become pyruvate. But if we target or get rid of CD36, we will have a different scenario. Those Treg will lose the PPAR beta signature, and they will not be able to maintain their mitochondrial fitness. And Treg will start to accumulate more lactic acid because they will not be able to convert those lactic acid into pyruvate in a sustained way. And they will start to die due to the high lactic acid condition in the tumor microenvironment. So with this information, we start to speculate, so maybe we should target CD36 to achieve this anti-tumor response. So we identify an antibody which can block CD36 lipid binding site. When we use this antibody as a treatment, we can start to see anti-tumor response. And similarly, we start to see intratumor Treg decrease, but the Treg in spleen and draining lymph node, they remain the same in the presence of anti-CD36 antibody treatment. And those Tregs, they're still alive in the tumor microenvironment. They start to show higher level of Cas3 staining. So it's also reflective of what we observe in the genetic mouse model. So basically, this data suggests that we can target this protein to achieve selectively, we can selectively demolish intratumor Treg by targeting this protein. So we next test whether we can use this antibody to achieve anti-tumor response in combination with anti-PD-1. So we use a very difficult, a mouse model which is very difficult to be treated. So this is the inducible BRAF PTEN melanoma system. And the entire infiltration is very low and the tumor is pretty cold and a lot of Treg accumulation in this tumor model. So you can see here, PD-1 treatment alone only induces a very minor anti-tumor response. And we can start to see a good anti-tumor response when we give them CD36 antibody. It can also be a little bit stronger than PD-1 alone. But importantly, when we combine them together, we basically stabilize the disease progression. So it suggests that actually we can use this antibody to selectively destroy intratumor Treg, which will allow us to improve the PD-1 blockade responsiveness in those tumors. So basically, I want to use the first part of my talk to tell you that we indeed identify a very special metabolic adaptation which will be used by intratumor Treg. And we also demonstrate that we can target this process to induce robust anti-tumor response. And the entire concept of biometabolic adaptation used by intratumor Treg has also been shown by Humboldt recently in his Nature paper. He also demonstrated that actually Treg in different tumor contexts can also engage lipid metabolism to support their survival. So I will move the gear toward to the second story, which is about CD8 T cells. So we also know that CD8 T cells in a tumor microenvironment, they will encounter multiple problems. They will not be able to maintain their function. And the major problem we have to deal with in the CD8 T cell is that those CD8 T cells, if they enter to the tumor microenvironment, they will start to upregulate PD-1. They will also change their transcriptome, and they will become fully exhausted T cells. And in this case, they will lose their functionality and also become non-responsiveness to immune checkpoint blockade treatment. And this becomes a major problem because those patients in this category, they don't show very good response after PD-1 blockade treatment. So however, if those cells they are in the partial exhausted state or in the progenitor exhausted state, they will be able to respond to PD-1 blockade treatment. And it has been shown those T cells, if they become exhausted T cells, this is the reason, the epigenetic reprogramming is the reason to drive this entire program. It's also a reason those T cells, once they become fully exhausted, they will not be able to activate it again by PD-1 blockade treatment because the epigenetic landscape already locked in a particular state, which will prevent their reactivation. Even the signaling cascade can be partially activated again. Despite we have this understanding, we still don't know what kind of mechanism can drive this epigenetic reprogramming. So if we can understand this process, we might be able to define a better way to sustain T cell anti-tumor response and also reprogram the CAR T cell we generate in vitro, because the same problem will also happen in CAR T cells. So my postdoc here decided to take a look whether the metabolic stress happening in the tumor microenvironment can be the one of the driving force to facilitate this epigenetic landscape change in T cells. So we first analyzed mitochondria. As I told you before, mitochondria is very important to maintain metabolic flexibility for any single given cells if in response to metabolic stress. So we isolate CD8 T cell from spleen and tumor from the same tumor-bearing host. And if you check their electron microscopy analysis, you can appreciate that those intratumor Treg contain a lot of mitochondria compared with splenic T cells. So basically, you can see the quantification look like this. They robustly increase mitochondrial number. So it looks very good. Actually, those CD8 T cells should be able to deal with metabolic stress happening in the tumor microenvironment. However, when we isolate those T cells from these two places and check their mitochondrial DNA, we don't see proportional increase. This becomes a problem because mitochondria always contain mitochondrial DNA. If we want to increase our mitochondrial number, in principle, every single given mitochondria will also duplicate their mitochondrial DNA in order to have a proper mitochondrial structure and proper mitochondrial activity. So in this case, we see more mitochondria, but we don't see mitochondrial DNA increase, suggests that those increased mitochondria in tumor infiltrating CD8 T cell, they may lose their integrity. They may not be able to operate properly. So indeed, that's the case, because when we use high magnitude to check those EN data, we start to see something like this. Although those tumor infiltrating CD8 T cells contain more mitochondria, most of their mitochondria look like the right-hand side. They lose the double membrane structure. They also lose the cristae, which is important to maintain membrane potential and oxidative metabolism in the cells. And we also perform 3D EN to double confirm the entire morphology of the mitochondria. So you can appreciate that each color here indicates one mitochondria. In a tumor microenvironment, you will see the mitochondria is more connected and more mitochondria. For example, you can see this yellow one is very connected. However, by using this 3D EN, we can further check whether their cristae start to show any problem. And we confirm that even they have more connected mitochondria, their cristae is also much lower compared with the splenic CD8 T cell. So basically, we can conclude that in contrast to intratumor Treg, CD8 T cell in a tumor microenvironment, they cannot maintain their mitochondrial activity. And in the analysis, we also noticed there are two different kinds of CD8 T cells in tumors. The first one look like the right-hand side. They completely screw up their mitochondrial structure. So you just see the structure, but they didn't look like the right mitochondria, as you can see in the textbook. And the other population you can see is on the left-hand side. You still see relatively normal mitochondria. They still have relatively okay mitochondrial structure and cristae. In this population, you can see this double membrane structure look like autophagosome. So we start to speculate maybe this population is the one they can engage autophagy and mitophagy to clean the damaged mitochondria. And we can also observe these two populations by using FACS analysis. We basically stain the T cells with two different molecular probes. The first one called Mitotrack green, which stain mitochondrial mass. The second one called Mitotrack deep red, which stain cells with mitochondrial membrane potential dependent dye. So you can see here, the one labeled in green, in this case, they contain more mitochondria because they have more Mitotrack green staining. However, most of the mitochondria decrease the membrane potential because they have lower deep red staining. So we can consider this MDR-MG low population is the population that accumulate a lot of depolarized mitochondria. And this population percentage increase in the tumor microenvironment compared with the CD8 T cell in spleen and draining lymph node from the same tumor-bearing host. And this is also holding true in patients, because once we get the melanoma patient samples, we can also see that majority of the patient we analyze in their tumor microenvironment, they will see higher percentage of MDR-MG low population. So back to the cell biology part. When every single cells receive metabolic stress, for example, low glucose, hypoxia, or in a condition they have to engage fatty acid oxidation, they will fuse their small mitochondria into a big one. In this case, they will be able to operate oxidative metabolism in a full speed. However, as you learn in the biochemistry textbook, if you want to run oxidative metabolism, you will also have a problem because you probably generate a lot of reactive oxygen species, and this will damage mitochondria. So this will accumulate a lot, then those cells will start to accumulate mitochondrial damage. But normally this is not a problem because they can get rid of the damaged part, degrade the mitochondria through the mitophagy machinery, which is a very special form of autophagy, and recycle the intact part. However, there are some of the cells, they will not be able to engage this process. They will not be able to engage fission and mitophagy. They will start to accumulate those depolarized mitochondria. This is very common in aging neurons, in aging muscle cells, and some of the aging immune cells like T cells. And those cells, they start to have this problem. You will see the sign like enlarged mitochondria and reduced membrane potential, which exactly what we observe in the CD8 TLs. And those the accumulation of damaged mitochondria actually has been shown to change cellular property and even induce senescence in neuron cells. So we start to speculate maybe those T cells in the tumor microenvironment accumulate depolarized mitochondria is due to the inability on engaging mitophagy. And indeed, that's the case. And I will not go through the data, but I will tell you that we also further confirm when the CD8 T cell infiltrate into the tumor microenvironment, they start to encounter a lot of problems to engage, to sustain their mitochondrial fitness. But in this case, once they lose their mitochondrial fitness, they will engage exhaustion process and lock them in a terminal exhausted state. So in order to confirm that, we have to know how to define terminal exhausted T cells. So when T cells become terminal exhausted T cells, they will have three major features. The first one is that they will lose their proliferative capacity. The second one is that they will start to sustain their inhibitory receptor expression, including PD-1, TIM3, and LEX3, even in the absence of the antigen. And third one is that those terminal exhausted T cells will have declined cytokine production, even in the antigen-free condition after restimulation, because the epigenetic landscape change lack the expression of these enzymes, these proteins. So to confirm they are terminal exhausted if they have a damaged mitochondrial accumulation, we engraft the cancer cells into naive mice and then transfer naive OT1. 15 to 18 days later, we can isolate splenic OT1 or OT1 TLs with different mitochondrial activity labeled here. The blue one is the one that accumulate a lot of damaged mitochondria. And then we isolate those cells and then transfer them individually into naive mice again. In this case, we can re-challenge those mice receive the OT1 with Listeria ova. So basically, we can use this approach to check whether the T cells they received have the ability to re-expand, have the ability to produce cytokines. So our data suggests that in those OT1 TLs, if they lose their mitochondrial activity, they start to have they start to accumulate depolarized mitochondria. They lose the expansion. They sustain the expression of PD-1 and LEX3. They also decrease their ability for producing interferon gamma in response to Listeria ova infection. So basically, this result further confirmed once those T cells in the tumor microenvironment accumulate depolarized mitochondria in the cytoplasm, they start to become terminal exhausted T cells. And we also confirm this is associated with epigenetic landscape change. So we first perform CpG island methylation pattern change. We start to see that those populations with a lot of damaged mitochondria, they display robust difference in terms of the DNA methylation pattern compared with the T cells in the tumor microenvironment. They still have good mitochondrial activity. In the ATAC-seq analysis, we can also see the same changes. Not only the DNA methylation pattern altered, their chromatin architecture start to change. And Among those genes, which will be largely altered once the cells with damaged mitochondrial accumulation, we observe that those cells with damaged mitochondrial accumulation, they start to have closed TCF7 locus and closed LEF1 locus. These two locus can control the stemness property of CD8 T cells. So basically, our data suggests that if the T cells, they fail to maintain their mitochondrial activity, they will start to display functional similarity to terminal exhausted T cells. And they also start to show DNA methylation pattern, chromatin accessibility change resemble to the terminal exhausted T cells. Most importantly, they start to lose their stemness property. So with this information, we also wonder, it looks like mitochondria fitness can control the property of T cells, but what kind of factors are those CD8 T cells in the tumor microenvironment encounter can drive the decline of mitochondrial activity? And we start to speculate maybe the T cell receptor signal is one of the driving force. So in order to confirm that, we perform the following experiment. We engraft the same cancer cells, but they express two different kinds of tumor surrogate antigen. On the left-hand side is the BRAF-PTEN melanoma cells expressing ovalbumin. On the right-hand side is the same cells, they express GP33, which can be recognized by P14 cells. So we engraft the same number of cells into two different flanks of the same mouse. We wait for 13 days, and then we co-transfer OT1 and P14 CD8 T cell into the tumor-bearing mice. As I told you, OT1 can recognize ovalbumin. P14 can recognize GP33. So we can see whether the tumor-specific T cells will accumulate damaged mitochondria faster or more severe than the other counterpart. And our data looks like this. DRMG low population is OT1. In contrast, in the GP33, P14 will display more in DRMG low population. And the quantification look like the right-hand side. It's clear cut. No matter which mouse we use, we always can see this trend. So it suggests that the soldiers which can recognize tumor antigen, once they enter to the battlefield, they will start to encounter this problem. But the bystander, they will have less chance to engage this mitochondrial fitness decline machinery. And we also wonder whether the affinity of the TCR versus the peptide MC class 1 is also matter. So we use the engraftment system I told you before, but this time we only engraft the BRAF-PTEN melanoma expressing ovalbumin. But we transfer two different types of T cells. One is OT1, the other one is OT3. Both of them can recognize ovalbumin peptide presented by the MC class 1. However, OT3 have 50 times lower affinity. As you can see here, if your T cell express lower affinity, they have less problem compared with the one have high affinity to the tumor antigen. So this results further suggests a very important idea. Your best soldiers, which recognize the enemy way faster, they will have problem to maintain their mitochondria in the real situation. So they will lose their mitochondrial fitness faster compared with the one they don't really recognize enemy that efficiently. And this is also reflected what we already observed in a single-cell RNA sequencing data in patient samples, because in human samples, those T cells with a lot of exhaustion features, they look like they have better tumor antigen recognition ability. So it suggests that we have to deal with this issue. Otherwise, no matter what kind of T cell you generate, as long as they have a good recognition ability to tumor antigen, they will fall into this trap. So as a result, I want to give you some idea that we confirm that the tumor microenvironment provides a particular signal and then coordinates with the TCR signal. They can drive this mitochondrial dysfunctional phenotype. And TOs with best tumor recognition ability tend to accumulate more damaged mitochondria. So based upon this idea, we propose this entire model. So we think that when T cells infiltrate into tumor microenvironment, in the beginning, they have very strong mitochondrial fitness. In this case, they can maintain their metabolic flexibility and anti-tumor response. They also sustain certain level of stem-like property. However, they will receive two different signals in the tumor microenvironment. One is hypoxia and chronic T cell receptor signal, and second one is glucose deprivation. And these three create a perfect storm. The idea is very simple. In a hypoxia condition like tumor, cells will start to use glucose. However, it is also a glucose deprived condition in tumors. So in this case, in order to survive, T cells will start to use oxidative metabolism in an error-prone way. They will generate too much reactive oxygen species due to the hypoxia condition, and this will damage their mitochondria. However, T cell receptor signaling will block mitophagy because TCR can activate mTOR. And as a result of this, those cells will start to damage their mitochondria, and they cannot remove the damaged mitochondria. So eventually, they will accumulate a lot of damaged mitochondria, and this will drive the exhaustion and change the epigenetic landscape, also reduce the cell's metabolic flexibility in the tumor microenvironment. So we also realize that we have to target this process. We have to find a way to sustain the mitochondrial fitness in T cells. Then we will be able to launch a robust anti-tumor response. So we use the in vitro system to introduce T cell receptor signal hypoxia and glucose deprivation by tumor cell conditional medium. We can induce the formation of this NDR and GLO population. By screening, we identify that nicotinamide riboside actually can also reduce it. And this is the one I told you before in the T-Rex story. This is an NAD precursor. It actually can also stimulate mitophagy because NAD can stimulate mitophagy. So basically, we found that you can use this same treatment, but in CDA, it can sustain CDA T cell number and reduce the mitochondrial dysfunctional phenotype. And we, in collaboration with Litan at EPFL, we put this nicotinamide riboside into liposome and do the intratumor injection by artificially provide those CDA T cell better stimuli that can sustain their mitochondrial fitness. So in this case, once we treat the liposome encapsulated nicotinamide riboside, we start to slow down the tumor growth as a single treatment. And in this case, we also sustain the CDA T cell stemness property and also sustain their mitochondrial fitness when we analyze those CDA T cell from the NR treated mice. So basically, I want to give you an impression based upon the data I show you here in CDA. Our data suggests that mitochondrial activity is very important to determine what kind of behavior CDA T cell will display. Basically, we learned that mitochondria, if they have a problem, they start to accumulate depolarized mitochondria. This will control their metabolic flexibility, so it will decrease the flexibility. At the same time, those cells with damaged mitochondria may change their extracellular communication because this damaged mitochondria will change in neuron or in muscle cell. We know that they will induce SASP, so this is senescence-induced secretin. And we believe that this can also be the case in the CDA T cells. They will completely change what kind of cytokine those CDA T cells start to express. And the other very important angle is that those mitochondrial damage will provide a different regulation to completely alter the epigenetic and transcriptome landscape in those CDA T cells. And you may wonder what's the reason mitochondria can do that. We have emerging, there are a lot of emerging studies from different experts in cancer metabolism and also immunometabolism speculate actually that so many different metabolites can do that. And I just provide you a summary, which was published last year from my group. We summarized those findings, and it looks like, for example, the SAM, which is a methylation donor produced by mitochondria metabolism, can control histone and DNA methylation. And lactate result from aerobic glycolysis can control histone modification in macrophage from lactylation. And very typically, the acetyl-CoA generated from acetylation or generated from fatty acid usage, and through the citrate-derived fatty acetyl-CoA, they can modify histone acetylation. So there are so many different metabolites can become the donor to modify histone or DNA. In addition, the metabolites in the TCA cycle, they can also serve as a cofactor or co-inhibitor to modulate epigenetic modifying enzymes ability. So overall, our data suggests that this crosstalk can determine how metabolic activity in immune cells control their behavior. So this will open a new paradigm for us to design the treatment and also how we should engineer those T cells we are producing in vitro. We may find a way to support their metabolic fitness in particular condition, and this will create a very interesting therapeutic window because we can control their activity only in the tumor, but we can reduce this kind of activity in the peripheral tissue. So overall, this provides a very important window for us for the following studies. And in the end, I would also like to thank people in contribute in those studies I just present, including Jet and also other people's in the metabolic angle, in the engineering angle. And most importantly, my team, without their support, it's not possible to conduct those challenging works and also the funding resource. And I'll be happy to answer your questions.
Great, thank you so much, Ping-Chih, for the terrific talk. So I guess if anyone has any questions for Ping-Chih, you can either type it up in the chat box, or you can just unmute yourself and ask directly.
I think Wuchuan has a question, right?
Oh, right.
Hey, Ping-Chih. That was wonderful. I mean, it's crystal clear. Really nice work. Actually, I just have two questions. First one, maybe I'm missing it. You're talking about antigen affinity, which actually regulate mitochondria fitness, controlling that the tumor progression. So I might just missed it. Is that the antigen stimulation in order to achieve that? The mTOR signaling pathway is the only pathway to control the mitochondria fitness, or their alternative signaling pathway?
So I think in this, so maybe I can answer this question directly before you say the second question. So I think mTOR might be the major one. The reason is that once you have the TCR signal, you will activate mTOR, right? And then the mTOR activation will suppress autophagy. And autophagy is the basic machinery for mitophagy. So we speculate this is the reason. So this might be this also might, I would say we don't have a direct evidence it works in this way, but the indirect evidence from viral infection also suggests this possibility. In a chronic viral infection, in the early phase of the infection, if you give it rapamycin, you will be able to sustain T cell mitochondrial fitness way better. And those T cells will have a better chance to become progenitor exhausted T cells. This is the study done by Exocadis in Immunity early this year. So they also support the idea that once they become terminal exhausted T cells, they lose mitochondrial fitness in a viral infection condition. But in tumor, it's hard to say because rapamycin treatment is too dirty. So we cannot really perform the experiment like that to confirm this.
But do you know it's dominantly through mTOR1 or mTOR2?
I don't know, but my personal preference will be mTOR1, but we don't know because get rid of the mTOR1 or mTOR2, the T cell also have some issues. So it's too complicated.
Right. The second question is, I saw the intra-tumor treatment with NR to stop the tumor growth. Actually, I think this is a question for general tumor or cancer biology, that because this treatment is more general, not systemic. I mean, normally you don't do such treatment on the patient. And then if you do general or systemic treatment, let's say in the case of, I'm not sure, infections or autoimmunity, will you actually see the similar phenotype? Let's say stop the CD8 T cell functionality, or in the tumor in general, in the systemic treatment, such NR injection, whether that will actually disrupt the CD8 function in general. Let's say, make the animal more susceptible to certain infections.
I don't think so. So the NR systemic treatment for NR is a little bit complicated. So we know that the NR treatment with the liposome is very effective to sustain CD8 T cell function in the tumor microenvironment. But we didn't try to do the systemic because the systemic treatment of NR, we have another cohort of paper in cell stem cell a few years ago. You will see a robust difference in terms of CD8, CD4, CD8 number. Yes, because the NR treatment, if you do it systemically, you will affect hematopoietic stem cell. You will make them younger, and they will have better chance to become T cells rather than the myeloid cells. So that's why we are not really sure whether this is a good idea, but it looks like NR treatment systemically can be a good thing. You will just robustly increase your immune system, but it doesn't facilitate autoimmune. So we don't know whether whether there's another threshold NR cannot pass. But basically, you just make them younger, and then they still listen to the normal regulation those T cell and immune cell need to listen to.
But with this kind of treatment, let's say if you do systemic in the tumor mice, what's the NR treatment in general will influence the tumor growth?
It will also reduce the tumor growth because your hematopoietic stem cell will be much good. They will do much better, and you will start to increase CD4 CD8 T cell count in the blood. So NR treatment looks like can be good. And NR treatment, once you pass to a certain dose, it also behave like a calorie restriction stimulating factor, and they will mimic calorie restriction to make the entire mouse behave much stronger in response to checkpoint blockade treatment, in response to radiotherapy.
Okay, so but let's say if you do such kind of treatment, whether the injection or the treatment are going to be one-time treatment, or you have to sustain the NR treatment for a certain time period.
Can you say it again?
I'm just saying that the treatment with the NR, you have to do that one time. You just need to do one-time injection, or you need to sustain the.
You need to sustain the NR systemic NR treatment. Most people do the diet. So in our hand, we do diet. We see the hematopoietic stem cell change. In Guido Grumo's data, they also they didn't use NR. They use NAN, the other precursor.
Yeah.
But they need to feed into they need to put into the diet and then feed the mice for a few weeks.
But in your intratumor injection, that's only one-time injection?
No, we inject every 2 days.
I see. So it's need to sustain the level. So
you need to sustain. Because if you don't sustain, the NAD level cannot be sustained.
So it will be metabolized very rapidly if you do only.
Yes.
I see. Okay, thank you. Yeah, cool.
Hey, Ping-Chih, I have a question. So I'm wondering, like these mitochondrial fitness change, do you see it only in T cells, like in CD8s, or do you see other tumor infiltrating immune cells, like myeloid cells?
So after our paper, there's another paper showing NK cells also have the same problem. So as you can expect, NK cells also start to decrease their mitochondrial fitness and then behave like dysfunctional NK cells. In macrophage, I haven't seen any report, but we know that macrophage will have the issue. Yes, and dendritic cell, I don't know, we didn't check the dendritic cell because the quantity is lower, so it's difficult to get enough cells to see.
I just like I was very intrigued by the fact that you showed that this whole like change in fitness is also antigen driven. So that kind of triggered a question whether, like, say, if the similar changes happen in myeloid cells, what would be the alternative signals?
I think in the myeloid cell, probably some cytokine activation is more important. But T cell, they listen to the TCR. The TCR is very strong signaling trigger compared with cytokine. So I think that's the reason in the T cells, in our setting, we can see TCR play a very critical role. But in myeloid cells, probably cytokine.
All right, thank you. I think Chaoran has a question.
Yeah, thanks, Chen. Great talk, Ping-Chih. So this is Chaoran from Emory. I used to work on adipose tissue Tregs in Diane Mathis' lab. So I'm really intrigued by your first part of your story, the CD36 part.
Thank you.
So my question is, basically, Hongbo also showed the fatty acid synthesis is really important for tumor Tregs. Yes. And your story is about the uptake. So do you know, like, what's the relative contribution of these two pathways, and can they compensate for each other, and why do you need both?
Okay, so I think, synthesis probably is very important for their expansion and also the suppressive activity. And then for the uptake, I think it's important for their survival. And there are two things we have to be careful about when we interpret those findings. The first one is that, we know that lipid metabolism is important, but we don't really know what kind of lipid we are talking about. We don't have the identity of the lipids. So it's difficult to say whether they get lipid from outside, they get lipid A, but they produce lipid B. It can be simultaneously operates because they need both actions. That's the first thing. And second thing is that we are not really sure whether the dependence of uptake and the dependence of synthesis depends on where those Treg they locate. Because we are talking about entire tumor, but in the entire tumor microenvironment, let's talk about the lectate part. you would not expect all the lectin level is always the same in different location. So the hypoxic region versus less hypoxic region, probably they will have different dependence on particular metabolism. That's something we don't have the ability to address at this moment yet. We probably will be able to address in the next few years with the special imaging system for the metabolomics. We will start to address whether the cells will also change their metabolism based upon how deep they enter to the tumor microenvironment.
Okay, thanks.
See, anyone else have any questions? I guess, Ping-Chih, I have one other question. So is the phenotype you saw tumor type specific? Would you see a similar phenotype in other types of cancer, or if the tumors are growing in a different microenvironment, in different organ site?
For CD8, I would say that it's more conservative, the mitochondrial defect, because we show it, and Craig Thompson also show it in the in vitro setting, and Greg Delgove used different tumor model also show it. And in the viral infection, people also start to show it. And then I haven't seen a paper, but I see people start to have the talk. In the HPV, HCV, and the HIV, they also see the same thing. CD8 T cells start to have mitochondrial defect. So it looks like this is very common. As long as the antigen stimulation is prolonged, you will see it. But the Treg, this part, the CD36, I think it depends on, we know that Treg will increase CD36 expression in response to some factors secreted by tumor cells, but we don't know which factors. So that's why I speculate this CD36 upregulation is tumor type specific. They probably can, in our case, they use CD36, but in other cases, they probably can use different fatty acid transporter. They don't really need to use the same one. Mm-hmm. They just need to use the same machinery. They can do it. Mm-hmm. That's what I would speculate.
Have you tested the other tumor models, not the YAM 1.7, but the other tumor cells?
So the CD36, we try MC38, B16, and YAM 1.7, we see it. But in human, it depends on the tumor types. We are still struggling and then try to define, because we also have I also create a spin-off, we try to target CD36 for treatment. So we need to find out the indication to know which patient we should use CD36, which patient we should not.
Great, thank you. So let's see if anyone else have questions for Ping-Chih. Okay, if not, thank you again very much. Thank you for the awesome talk, and thanks everyone again.
Thank you, everyone. Bye-bye.
Take care.